So I think we'll just start. So welcome back to the seminar series by the International Tree Mortality Network. So it's a great pleasure to welcome Kimberly Novick from the Indiana University in Bloomington and Jessica Guo from the Harvard Mudd College. So they are both ecophysiologists who are co-founders of the PSInet, which is, so please correct me if I'm wrong, intended to counteract the global water potential gap. I assume that the ecophysiologists in the audience are particularly aware of the importance of continuous water potential readings for understanding the drought response of plants. So I'm really curious to hear about your progress. And with that, thank you already now for agreeing to present today. The floor is yours.
Okay, thank you so much, Bernhard. I'm really grateful for the opportunity and really grateful that this webinar series exists. I think it fills a really important niche in sort of the broader ecophysiology, plant hydraulics world. So thanks so much for having us. Let's see, I'm just trying to move my toolbar so I can see. There we go. So this talk is largely going to be about a database of plant water potential time series, which we are getting ready to release. But I'm going to take a little bit of a meandering route before we get there, just sort of doing some stage setting and taking care of a few preliminary things. The first thing to note is that the database is a product of a research coordination network called PSInet. Though if you prefer to call it CNET, that is just fine. Anyways, this is it's a project more than just a database. It's funded largely by the U.S. National Science Foundation. And we have three really primary goals. The first is support consistent water potential data collection through best practices training and other centralized initiatives. The second is to build databases of plants and also soil water potential time series. To support then the third objective, which is effective end use of these network databases to address questions that just simply would have been harder to address if these data were not available and accessible. And a lot of that effort is focused on workshops and building collaborations with other environmental observation networks. There's a lot of people involved, and I feel I'd be really remiss if I did not acknowledge them before we get to the science. So I first want to acknowledge the co-PIs and key personnel on the project. And in particular, I want to thank Kate McCullough, Dan Johnson, and Jesse Nippert, who played crucial roles in helping us meet that first objective, supporting consistent water potential collection by organizing a couple of early career workshops that happened over the past few years. The first was the 2023 PhysFest workshop in New Mexico, and then also a 2024 workshop in plant hydraulic methods. Each of these was quite successful, reaching dozens of students and training them through really hands-on experience with a range of ecophysiological instruments. So here's just a few images from PhysFest, and it's my understanding that PhysFest is happening again soon. We sponsored the 2023 iteration. I think that the current iteration is likely sponsored directly by NSF. It happens every 2 years, and it's a great opportunity. So keep your eye out for opportunities in the future. My suspicion is applications are closed for this year. We also had a couple of committees, in particular a steering committee and also a data committee that were really instructional in the early phases of the project, helping us scope the goals, helping us write the proposal, and also helping us with the design of sort of the cyber infrastructure of the database. So I want to be sure to acknowledge those folks. These folks also played a critical role in soliciting data submissions once we were ready for that phase of the project. We have a number of what we'll call data scientists or just data wranglers, people that helped us organize the data as it started to come in and to convert it into accessible formats, which Jess will talk about halfway through the presentation. And I realize I'm remiss here. I should certainly have Ana Maria Restrepo Acevedo listed in this group. She was instrumental in helping us build the database. We also benefit from a lot of coordination with collaborating networks, and in particular, I want to acknowledge SAP-FluxNet, who was really helpful in designing the database submission template, among many other favors. And then finally, we had a workshop last year, and I want to acknowledge the organizers of that workshop, which was really useful for thinking about what we will do with the database once it is published. Okay, so with that, Oh, here's a few pictures from our workshop, which was held at Indiana University in Bloomington. So the major motivation for the network, for the project, for the database recognizes that on the one hand, thanks to remote sensing information and weather and soil stations, we have a lot of information across a range of scales of the drivers of plant function, right? The weather data, the changes in radiation and precipitation and vapor pressure deficit that ultimately determine plant growth and carbon uptake and survival. Thanks to what has really been an explosion in environmental observation network data availability, we also have a lot of information about plant responses to these drivers. But what is missing, or historically has been missing, is accessible information on the time series of water potential that connect the drivers to these responses and that move water through ecosystems and also, of course, directly affect many aspects of plant ecophysiological function. So this is the data gap that PSInet was created to address. And I'm going to return to this slide in the second half of the talk. Each of these topics could probably be its own webinar, so we're just going to touch briefly on them. But there is, a lot that we could do with more accessible, more continuous, and more representative water potential time series. Okay. So like I said, most of the talk is going to be focused on plant water potential and our new database of plant water potential time series. But before we get there, I do want to say just a few things about soil water potential, because of course they are not disconnected. So this looks like a slide from maybe Soil Physics 101, but I think most of us in this community understand that soil water potential is the fundamental driver of soil water flows. The difference in water potential from one point to the next is an energy difference that drives the bulk transport of fluid. For this reason, soil water potential shows up in fundamental equations like Darcy's law and Richard's equation. But for a number of reasons, it's not a variable that we often measure in the field. It used to be really hard to do it. It's probably the primary reason. So instead, we tend to measure volumetric soil moisture content, the volume of water per volume of soil, which is a useful metric, but it is not soil water potential. The relationship between the two tends to be quite nonlinear and frequently threshold driven. This is some examples of the water retention curve for 3 different soil types. So of course, differences in soil texture, but also structure will affect the shape of this curve. And do note that the y-axis on these panels is log scale. So if we want soil water potential, but we haven't measured it, instead we measured soil moisture content, a common thing to do is to apply pedotransfer functions to predict water potential from a given soil moisture content. And I suspect that anybody who's ever worked with these in a really hands-on way knows that they are quite uncertain. In fact, small tweaks in just one parameter of a very popular pedotransfer function, this is the Rosetta model, can produce orders of magnitude scale uncertainty in our guess at soil water potential at a given soil moisture content. And it turns out that this uncertainty linked to pedotransfer functions is actually quite a big source of uncertainty in terrestrial process-based models. So this is the result of a sensitivity analysis performed by now a few years ago, Nina Raoult, where she was working with the Orchidea model, and she took all of these really crucial parameters in the model and tweaked them a little bit this way, a little bit that way, and then quantified the extent to which GPP was affected. And the take-home message is that the water retention curves parameters in blue and the soil hydrology parameters more generally produced a lot of variance in GPP, even with just small tweaks. So in other words, our models for GPP and related variables are very sensitive to even small changes in these parameters. This also, affects our ability to conceptually understand observed responses. It's a very common thing in our field to put on the x-axis soil moisture content, and then to put on the y-axis, your favorite flux or conductance, maybe GPP or surface conductance or stomatal conductance or transpiration or what have you. And oftentimes these relationships tend to be nonlinear and apparently threshold driven. And so one of the hypotheses that we are hoping to test is the notion, that a lot of this nonlinearity can actually be explained by the shape of the ecosystem water retention curve, such that if we are simply able to switch out the x-axis from soil moisture content to soil water potential, we should see these responses become more linear and more homogeneous in space. Okay. This is an idealized scenario. Of course, there are many reasons why we still might see threshold type responses, but we'd like to get rid of what is perhaps an obvious source that maybe clouds our view on what else might affect the relationship between these variables. Okay. So how can we address this water potential gap in the soil? Of course, we can measure it more. And truth is that the instruments for in situ, at least metric water potential measurement, have come a long way over the years. We like to use this one produced by Meter Group at our site in Indiana, where it produces pretty tractable and sensible results. However, this one really doesn't work very well in dry soils. So if you're working in a more arid ecosystem, you might need a different sort of instrument. And so there is still, I think, a need for community developed protocols, guidance, and best practices for in situ measurement of soil water potential. And I can say that I field a lot of questions from PIs just asking, so what do you think is the best soil water potential to use at our site? Okay. I also want to take a minute to highlight this project, which is ongoing. It's the Regional Environmental Soil Hydraulics, or REESH project. And the goal here is to generate water retention curves, that relationship between soil water potential and soil moisture, in the lab for ecophysiological and ecohydrological research sites. Right now, it's focused primarily in the United States, but we do have the ability to accept international soils. And so that's something that we might look at into the future. It's very simple. We mail out sampling kits to PIs who then go out to their site and collect 12 cores from 3 different depths at 4 different sampling locations and send them back to us, where they are run through this gamut of soil hydraulic property measurements. This is a project that was largely designed and implemented by Alex Crookshanks, formerly an IU undergrad, now a PhD student at Columbia University, and Daniel Beverly, formerly a postdoc at IU, now working for Meter Group. Okay, I should note that this map is a bit out of date. I didn't have time to update it before the talk, but at this point we have 45 sites fully processed, and so you can download and use the data for your research freely. Just check the data acknowledgement policy if you do. We have 11 sites actively being processed and 12 sites in the queue, and hope to keep this going as long as we can. So we're always looking for more sites that want to be integrated into the project. We have submitted our first paper emerging from the REESH project. It was led by Alex with Daniel as senior author. And this is reporting results from maybe the first 18 or so sites that we processed, which do a reasonable job of covering the soil texture triangle, though of course there are some gaps. And I'm just going to show you 2 key results. The first is that there can be quite a bit of difference between the water retention curves that we measure in the lab and those that we might predict absent that lab-based information. So this panel is the water retention curves predicted using a gridded soil texture product and the Rosetta pedotransfer function. In the middle are the curves, again using the Rosetta PTF, but now with local measurements of soil texture. So even measuring soil texture locally can produce quite a bit of divergence from what we otherwise would have predicted. And then here are the REESH lab-derived curves. So there is certainly a difference. And then the other thing I want to show you is just that when we use the REESH curves as compared to these other approaches for predicting soil water potential, we recover a higher correlation between latent heat flux, which is evapotranspiration, and soil water potential, compared to the other approaches, and also compared to the relationship between latent heat flux and soil moisture content. Meaning that, that hypothesis that if you put soil water potential on the x-axis instead of soil moisture content, the curves will become more linear, is confirmed, at least with these early results from the REESH project. And I also just want to mention, put in a little plug, that this is part of a ongoing NASA-funded project being led by Julia Green at the University of Arizona. The goal of this project is very simple. It is to transform SMAP data into wall-to-wall estimates of soil water potential, beginning with the continental United States, but we hope eventually we can find a way to expand globally. So it's very much a work in progress, but it is exciting. So stay tuned and keep in touch. And as these products become available, we hope that you use them in your research. So with that, of course, there is another way to get at soil water potential, which is the trick that ecophysiologists have been using for decades. It's the assumption that in pre-dawn hours, water potential is equilibrated throughout the ecosystem. So that pre-dawn measurements of plant water potential give us at least a pretty good guess, with some uncertainty, of the soil water potential across the root zone. So with that, I think it's time to move on to the plants, with the acknowledgement that there are many more reasons, of course, to be interested in plant water potential beyond its use as a proxy for soil water potential. And here, the issue is not so much that we don't have the data. For decades, since the 60s, I think, scientists have been measuring water potential, usually with this device called the pressure chamber. There are thousands of time series of plant water potential in existence, right? A quick Google search for Google Scholar for pressure chamber or Scholander or plant water potential can confirm this quickly. But the issue is that historically, these time series have been unnetworked and undiscoverable, right? There is no place that you can go to get a lot of water potential time series at the same time, okay? So that is one issue. Another issue is that the existing pressure chamber data tend to be characterized by variations in protocols and measurement techniques from one site to the next, which can hinder synthesis. And as we were writing the proposal that funded PSInet, this paper came out, and I was really grateful for it. For on the one hand, but basically reviews and issues best practices for the measurement of leaf water potential. Which meant we could cross that off the list of things that PSInet needed to do, but also this paper was really useful for informing the metadata of our own database so that we can be sure that we can track how those protocols vary from one time series to the next. So it's a great paper in case you haven't stumbled across it already. Another issue with pressure chamber measurements in particular is that they are hard work. It is a manual and destructive measurement, especially if you're doing those pre-dawn measurements. It results in a very early alarm clock. And, I don't want to say that the only reason that we pursued the PSInet idea was to avoid having to do pre-dawn measurements in the future, but it certainly was a motivating factor. It's really hard to get up that early and do all this hard work, especially if you work in a tall forest canopy, as we do, where you have to have all of these tricks to get to the top of the canopy. And so, it's as a result of the fact that this is so destructive and labor intensive, the temporal resolution of these time series tends to be relatively low, maybe weekly, maybe monthly, which is often sufficient to understand plant responses to drying soil, but is too coarse to understand how plants are responding to more rapid fluctuations in vapor pressure deficit and air temperature. Fortunately, over the past, decade or so, we've seen a lot of emerging approaches for continuous in situ monitoring of plant water potential, including psychrometers and also more recently microtensiometers. And this is the point of the talk where I am going to hand it over to Jess, who is much more of an expert at the use of these data than I am. So Jess, just tell me when to advance the slides, but
All right.
Yeah.
Sure thing. Next slide, this is a replay. Just want to reiterate that the pressure chamber is still is the gold standard. So in my experience using the psychrometers, I haven't really reduced the number of pre-dawns taken. But, the information content is just very different, just to reiterate Kim's point. So here, this is from my 2020 paper, last chapter of my dissertation. The pre-dawns here tell you some information, right? This is for a desert shrub, creosote bush in the Sonoran Desert. And you can see the water potentials are high in the spring, drop down, respond to some monsoon precipitation, drop down again, and then recover as the winter rains come. The advanced slide. And then if you fill in what the cyclometers are doing, this is half-hourly data summarized to pre-dawn and midday for six shrubs at a time, 12 shrubs in total. You can just see there's just so many more smaller events that are captured in addition to the soil moisture drawdowns that you can really now start to dig into the effect VPD has on water potential. So as far as I am aware, I don't want to claim to be the first, but this is one of the earlier like water potential time series outside on like a naturally occurring plants. Advanced slide. The next iteration, this was during COVID. Steve Kannenberg, got some. He was a fantastic collaborator. Got some money to put up this flux tower in southeastern Utah, and then the idea was, okay, there's already a tower here. Let's try to put some cyclometers in. Let's try to get compare what the fluxes look like with the plant water potential directly. So again, the top panel here is the half-hourly data. This time on juniper, Juniperus osteosperma. Summarized to pre-dawn and midday. The brown points again are the chamber measurements. This time, I think Steve did not quite want to get up for pre-dawns to validate, but hey, you can get up at a different time to validate. You still have the cyclometers are still measuring during that time too, and they match pretty well. So that it gives us confidence that the cyclometers aren't just measuring something spurious. And then you can compare it to the bottom panel, which is GPP, and wow, we really can see that like this year, this is 2021, there were these 3 major monsoon storms that hit this far north into Utah, and we can see the really immediate responses in the plant water potential, and that there are these somewhat lagging but slowly less lagging responses in the GPP as the plants get kind of awakened or activated again after after the pre-monsoon drought period. All right, so we learned a lot from this, just from doing this a bunch and like having lots of frustrating experiences and troubleshooting and then trying again. We learned a lot about how to use cyclometers, and I was able to pass that on to Steve, for example. But it was less effective than, next slide, kind of putting all of our heads together, because I only use cyclometers. They work really well on the dry end. You can see some of those water potentials, pretty negative. They work well on the dry end. They're not sensitive and tend to get more and more artifacts when you get to the wetter end. There's a different technology that Kim showed earlier, the microtensiometers made by Fluoripulse. And but I don't have personal experience of that. I have to hear about it from other people. There are in fact a lot of folks on this author list here, Sharath in particular, Ruth Christina come to mind, who are really experts in using the experts and generous in sharing their knowledge of how to use the microtensiometers. If we put our heads together, Anna Maria led this nice paper that just was available, I think, just a day or two ago, a new phytologist on the methods of how we can advance and use these continuous monitoring approaches and what the factors kind of keep in mind. These are expensive sensors. They can break. There is a lot of labor involved in going out and checking on them and like troubleshooting them. So now I feel like I'm excited that there's a first paper out on how to even strategize for your species, for your site, which kind of approach might work best. Okay, next slide. But okay, I'm also here to talk about the PSInet database. This has been a really huge effort by the community, and I've been really astounded by the generosity of people sharing their data. There's quite a bit of unpublished data in the database, people who are just willing to just be like, here, have it, and we'll share it with the world. So that is like really one of the main key feature is there's this global database. Maybe it doesn't sound terribly huge in this era of big data, but of course, many of these data sets were manually collected. And so it represents people's actual, physical labor and getting up at pre-dawn and shooting the trees or climbing the trees or whatever to get it. So I really appreciate everyone's generosity there. We have both pressure chamber and automated data. There's fewer automated data sets, obviously. It's a much newer technology. We've thought very deeply about the metadata and what to document. We asked people to contribute soil and metadata. Not everyone did, of course, that wasn't always available. But those gaps can be filled in with gridded products, for example, or other methods. We also really prioritized connections to other networks and databases, including first and foremost, we're kind of built upon the architecture in some ways of SAFFluxNet. They came first. They were like really, steering committee members, early cheerleaders, shared a lot of their data and protocols with us. There's also a lot of connections with FluxNet. I was pretty surprised. The automated data especially has a lot of overlap with FluxNet sites. By the time you already put in a tower, it's like, okay, well, what else can we do? Maybe putting in continuous sensors is a good next step. XFD, that's the Xylem Functional Traits Database, currently managed by Bill Hammond, and Riesch, of course, that Kim talked about. All right, so let's go to the next slide here. I want to share kind of what our data ingestion process was. So first, I just want to really say that this was a human-led process, right? Like taking a water potential measurement, especially at pre-dawn, involves, stumbling around in the dark, reading your pressure chamber, trying not to run out of gas, writing down some numbers, maybe in a notebook, and then eventually putting it into the computer and then giving it to us in a template. So it's just a very human process. So although it is script based and reproducible, everything's on GitHub for every single script that came to us, how we fixed it so that things are traceable. It was a very human-led process. All right, and so we all know how water moves through plants going up. So I made a diagram that follows this. We had our PSInet template. Our data committee helped us think deeply and beta test like different versions. Template, of course, later on, there were a couple times where we forgot something and had to go back and ask for it again. Could not be helped. And our data came into this data template via both SAP FluxNet. They had already asked some of their members for water potential data that accompanied their SAP flow data, and they were generous enough to just port that right over, so we converted their template into our format. We also had a large number of direct submissions. Okay. The first step was a QA/QC step by the data science team. This was, largely scripted. We were just this was a multi-tab Excel template, and then we imported it tab by tab and checked for different kind of internal compatibility units, time series. It was a time series, that sort of thing. We defined time series very generously. 2 points is a time series. Okay, and the next step, though, was to create a proof. We checked for the most obvious things, and we corrected the most obvious things, like typos or spelling errors. But we also had to send data back to the data submitter so they could approve, A, both of our changes, and B, add any additional required info that was missing from the first time around. Once they did that and turned it around to us, the next step was this validation. We conducted a final set of checks to make sure that the different tables joined with each other properly on the we had everyone's authorship and DOI information. We created a set of flag tables with some range checks. And so that was that. All of these sheets that people gave us are turned into relational database. So this, it happens to be in a DuckDB format, but it's available to anyone who can use a database, use their language of choice or a database access tool of choice to manipulate this database. But I'm an R user personally, so there was also we also made an R package. All this R package does is it downloads the database locally, and then there are a few custom joining functions that pull these different tables together into what I think of as like analysis-ready kind of tables, data frames in R. That's all it does. If R is not your choice, though, you can either use, access the relational database directly, or we're going to be putting, this is not there yet, but we're going to be putting all of these flat tables for the whole database on Zenodo, and that's just going to be stable and accessible as well. In case you're interested, next tab. We have kind of different GitHub repositories for each of these kind of steps where you can follow along what we're doing. Okay. So that's all the process. What's in the database? I think this is a key time to talk about this. So we have a total of 284 data sets, the vast majority of which are chamber data, 16 of which are automated data. And yes, some data sets had both chamber and automated. This map on the left here, the top shows where all the chamber data sets come from. Notice the concentration in the usual locations, North America and Europe, but also we do have quite, some coverage of South America, Australia. Africa's not looking that bad, actually. A little sparse in Asia. The automated data sets, the size of the circles here are by number of observations. So the circles are bigger because automated. And they are also, there's only 16 of them, but they're distributed there on the landscape. All right, next slide. Just as a comparison here, the top panel here is the number of data sets, where the number of chamber data sets is greatly outnumbers automated. But if you look at the number of observations on the bottom panel B, the number of observations, again, from the automated outstrips the number of chamber. And that, again, speaks to the labor and intensiveness of those chamber observations. All right, next slide. I also want to highlight the colors here. I haven't talked about those yet. These colors indicate the overlap with other networks, whether that's some kind of covariance network, whether FluxNet or a regional network. SAFFluxNet, which is abbreviated here SFN, or both. And so we have quite a bit of overlap there, especially in the automated data. All right. Next slide. So this trade-off that this like flipping that we were seeing in the previous slide, I want to investigate that further. And so if we look at the trade-off between measurement frequency and measurement duration, and the chamber data, that's very pronounced. So here on the x-axis is a log measurement of kind of measurement frequency, water potential per plant per day. And the y-axis is just divided into bins of duration that seem to make sense based on the kinds of data that we had. And you see this pattern, this linear kind of trade-off. But this excludes the orange points here, which are the automated water potentials. And you can see there that, we really, this trade-off can be broken with the automated data set. Just not to say that there's no labor involved, but it's just, the machine can keep working when we need to go home and sleep. So, all right, next slide. Okay, this is I'm really proud of this figure. We ended up with 517 species in this database. And this was kind of a last minute request for plant functional types, which is not my forte, not my domain. Turns out it's actually not super easy to translate like biological information about taxa into these categories that are useful for more modeling applications. But we have a lot of tree representation, shrub representation, not doing too bad on the forbs herbs. We do have a somewhat paucity of grasses, but not too shabby. And then, for pteridophytes, so ferns are pretty minorly represented. But we have a lot of trees and shrubs in this database, as you might have suspected. Next slide. Here, I just wanted to show the top 10 species on the top here for chamber and on the bottom for automated observations. Again, lots of trees. Quercus ilex is, I think, our #1 most measured tree in the database, at least for chamber measurements. There is evergreen shrubs in the bottom panel that is not present at the top. That's thanks to many measurements of water potential on creosote bush. But again, trees and shrubs mostly represented. I don't think this is a gap. There is a gap in the automated measurements of forbs, herbs, grasses. I'm not super aware of how to do that. There might be some species that have a wood-like or stiff enough stem that the psychrometers or perhaps the tensiometers could be installed, but I'm not aware of like leaf psychrometers, for example, that are advertised for leaves, like those really being able to be kept outside. So that is a gap. All right, next slide. Okay, I'm really excited about this. Sorry, this was not more jazzed up, but we were able at the end here to collect kind of like the most awards. And so lots of people to acknowledge here. There's the folks with the most chamber and automated measurements, Laura and Steve. Our longest time series is over 25 years. This is Brashears, McDowell, like Brashears et al., basically, the Pinion and Juniper data set that kicked off a lot of kind of interest in this kind of work. Associated with mortality, of course, the tree mortality on the Colorado Plateau after the 2002-2003 drought. Largest number of individuals measured. This is, I believe, a European data set. Number of species measured was out of Australia, Jen Peters and Brendan Choate. Field sites, etc. And I do especially want to acknowledge here at the bottom are two, like our largest number of cross-network coordination and studies by submitters. So Jordi and Rafa are both leads on the SAFFluxNet project. They also contribute a lot of data personally and cross-linked with SAFFluxNet to our data set. So really a lot of our, this database is theirs as well, basically. And then finally, this data set, the longest data set with data from both that is co-located with a flux tower comes from Joe and Dennis Baldacci. This is the Tonzi Ranch. And that spans about 20 years from like early 2000s to like 2023. It's also really cool. All right, next page. Also wanted to recognize not just the most, but also the firsts. So these are some countries for which there was only one, or non-European, non-North American, non-European countries with one data set submitted. So we have the first data set from India, Israel, Namibia, New Zealand, Russia, Senegal, South Africa, and Thailand. And also, I just have to recognize this country because it came in at 2 rather than 1, but we have 2 data sets from Cameroon, first by George Koch in 1991, and the second by Lucette and Rafa in 2025. So a large gap in between there, but Cameroon is represented twice, which is pretty cool. All right, next. Hopefully, we've excited you now and inspired you to want to work and look at these data. Who has access and when? So we have these like 3 kind of products we're going to have. Currently, what's available is the PSInet DB and the PSInet R package. And currently, workshop attendees and data contributors have access to those 2 formats. It does require a GitHub handle. That's how we're controlling the access to the PSInet database. If you are an author, we will be a data contributor, and you haven't heard from us in a while, fear not. As Kim said, we're working on that paper, and the next stop of that paper is to go out to all the data contributors, all of our co-authors, and at the same time, we'll be asking, do you want access to this? If so, please give us, the data GitHub handle, and we'll get you access so that you have early access to the database as promised. Then the next step on the timeline, unclear of exactly when that will be, of course, depending on the review process, is the publication of the data paper. And then 1 year after publication of the data paper, that's when we are going to be releasing all 3 of these versions to everyone under CC BY 4. All right, I think I have 2 more or 3 more slides after this. So the next one here, back to the 5 kind of what things could we do better with a centralized database of water potential. I'm going to focus out here on the first one. So I think next slide. Yes, of kind of advancing the theory. One thing that I was, that I've looked at in Creosote was how the water potential strategy, this is the Martinez-Vialta metric of the slope between the midday on the y-axis and the pre-dawn on the x-axis, including what's shown here, how that changed within a species in creosote bush. And that was, detectable with the long time series that I had with the psychrometers. But at the time, I remember people asking, well, maybe that's just creosote. Like creosote is a pretty unique, weird, drought-tolerant desert plant. Like just because it happens there doesn't mean that it is something that happens in more places. I didn't have an answer then because I hadn't measured water potential much in other species at that point. But with a database like this, that is exactly one of the questions that we can tackle. So the top row here, we're finding out, A through C here are some of the most popular, commonly measured species in the database. I believe these are all in Europe, actually. And so we colored these lines by different studies, and we could find a range of the slope between midday and pre-dawn across the different studies. Now, whether that's attributable to different times of year or different local topography or soil conditions, or maybe different years, drought years versus not drought years, we haven't delved in that deeply, but we do see that there is variation in the slope in non-creosote species. The bottom row, I think, was even more interesting to me, was that these are the same species in the same study, but in different treatments. We also had studies that had imposed experimental treatments, whether that was indoors in a greenhouse or outside. And so the D panel here, Digitera californica, is a C4 bunch grass, and based on the irrigation interval, the slope seems to change. This Pinus sylvestris in panel E, came from a drought experiment, and the slope there seems to differ based on the drought treatment. And then finally, this F, Quercus geariana, comes from, these are not even experimentally imposed treatments. They set up plots in areas where the oak was being not invaded, moderately invaded, or heavily invaded by Doug fir, and the slopes there also appear to be different. So that is just a taste. These are admittedly cherry-picked from the database so far. This is what people gave us, and these were the treatments they gave us, and we were taking a look at these. But I think it's it's a nice early hint of what's possible with the water potentials database. Okay, Kim, back to you.
Yeah, thanks, Jess. And the few minutes we have left, just going to run through a few more potential applications of the database. And I just want to say that when you see the word "we" here, that is the royal we. We is not me and Jess. Maybe Jess will write that paper. She just maybe not. But the hope is that these are figures that'll be part of the paper that will publish to accompany the database that are to inspire more detailed work by the community. So once the paper is published, within a year, everybody will be able to access the data openly. In that first year, however, the data contributors, and there are a lot of them, will have access. So perhaps you are one of them, or one. Anyways, the point is to sort of engender end use by everyone. I want to say a few words about vapor pressure deficit, which is one of the factors, in addition to others, that might explain some of that variation in the sigma over time. The theory and models suggest that vapor pressure deficit should be an important driver of plant water potential. So here's a figure from this paper led by Steve a few years ago that just shows the range and variability in midday water potential as VPD rises, even as soil water potential is held constant. However, in the absence of the right data, most of our inference about the role of VPD in governing water potential has had to be coming from modeling studies. If you have continuous data, though, you can do more. So we're going back about a decade now to a paper that just made the point that while soil moisture and vapor pressure deficit tend to be tightly correlated over long time scales, in fact, at the hourly time scale, they are relatively decoupled. Soil moisture doesn't change that much over the course of the day, but VPD can vary quite a bit. So it's possible with these oversampled high-resolution data to do empirical approaches, statistical approaches. This is an example of a data binning approach to just sort of bin the data by soil water condition, and then within each bin, assess the relationship with VPD. So this is sort of a schematic for how surface conductance might look as a function of VPD as soils move from dry to wet. In a site where there's relatively little soil moisture limitation, these lines might be very close together. In a more arid site, they would start to spread out. So we can do things like this with data like flux tower data and sap flux data. And here are just a few results from a few representative flux tower sites showing that, yes, for this very wet site in the Pacific Northwest of the United States, there is very little soil moisture limitation to surface conductance. However, over here in this desert, I think it's a shrubland or a grassland, we see the crucial role of soil water potential. With the continuous water potential data from the psychrometers and the tensiometers, we can begin to do similar exercises, but now with plant water potential. So these are just two representative figures. They're the same idea, just binning by soil water potential, and then exploring the relationship to vapor pressure deficit. On the left is Jessa's creosote site. And I think it's kind of interesting that the slope of this relationship is relatively invariant as soils move from wet to dry, but we can compare that to a situation for this oak species, where when soil is wet, there's relatively little influence of VPD on water potential, but then as soils begin to dry, we see that elevations in VPD drive down water potential even substantially more. So this is just a flavor. There's certainly a lot more that can be done there. A couple more quick things. I want to say a word about predicting the risk of hydraulic failure, which is usually estimated as something like the hydraulic safety margin, the difference between, the minimum plant water potential and some metric describing xylem vulnerability to embolism, let's say P50. And historically, we've gotten most of that information, at least for synthetic studies, from databases like XFT. And thank goodness for XFT, and we've learned so much from it. But the challenge is, particularly for minimum plant water potential, what you estimate as the minimum plant water potential depends on when you made the measurements, right? If you make your measurements in a period of a couple of relatively wet years, you'll have a much higher value than if your study period included a drought. And so what you see here are is a figure showing some data from PSInet, where the same species was represented across multiple data sets, multiple sites. And so each dot is that data set specific estimate of the minimum water potential. The purple square is sort of the average across all of those data sets for each species in PSInet. And the triangle is what's available for those species in XFT. So the good news is there is some correlation here. It's not just, which is encouraging. But with PSInet, we have an opportunity to dig a bit deeper and perhaps refine some of our estimates for the species level minimum water potential based, on data that may capture a more representative set of environmental conditions. I also just want to highlight this paper that came out a few years ago that describes specifically the use of extreme value theory for estimating minimum water potential. It's kind of the same thing engineers do when they need to know what is going to be the 100-year flood event, but we don't have 100 years of data. Just applying these distributions that are designed to capture extreme events can help us make maybe a more, as they say, robust determination of minimum water potential. But to apply this broadly, you need the data. You need the time series of water potential, which again, PSInet can provide. So somebody should really look into that. And then finally, I am not the right person to talk about either of these topics. So I will just say very briefly that, of course, there's a lot of opportunity to integrate not only PSInet data, but PSInet data together with StatFluxNet, together with Flux Tower data, into either assessments and benchmarking and parameterization of process-based models, or to validate remote sensing proxies, for example, vegetation optical depth or VOD data, which tend to measure canopy water content, but with the right sort of pressure volume curves can also be used to estimate water potential, right? So in this case, we imagine that PSInet data could be an important source of validation data for these remote sensing products. Whether it's modeling or remote sensing, I want to just emphasize that both applications will benefit from data, not only from PSInet, but from these other networks too. And both of them require careful approaches for scaling, right? From organs to plants to the ecosystem. Most of the data in PSInet is organ level. And here I think that the PSInet database allows us an interesting perspective on strategies for scaling. So what you see here are a range of sites in the PSInet database that have at least 3 species measured. And so each of these dots is sort of the average and maybe variability in water potential for each of those species, right? So the question is, each of these sites is, well, how do we, what is the best averaging or upscaling approach to move from these individual species up to the ecosystem? And, in some sites, it looks like it'll be pretty easy. These dots are kind of all right on top of each other. In other sites, it will be more challenging because we see quite a lot of inter-site species-specific variability. I think there's really rich opportunities for some people to really get a best practices for scaling in this way is, I think, a much needed contribution to the literature. In case anybody's been thinking about it and thinks, oh, maybe I'll write a paper about scaling from the plant to the ecosystem scale, strongly encourage you to do that. I think it is really needed. So we're done. Again, the PSInet database will be published in the coming months. We hope that you will use it to do some cool science. That is the main message of this talk. Maybe you have to wait a year for it to become open access. Maybe you can make a friend with the data contributor and get access earlier. And this was just a flavor of some of the applications of the database. Many of you all might have even more creative and exciting ideas. And of course, we are very grateful to the hundreds of scientists that generously shared their data, to the many scientists that helped guide the evolution of the project, and huge thanks to NSF for being the primary sponsor of the work. Thank you.
Yes, thank you very much, Kim and Jessica. Highly impressive and important work. I think we have a community should try to motivate everyone at least to contribute published data. And I think people are somehow scared a little bit. So I'm kind of impressed if you also received quite many unpublished data. But I think at least published data should always automatically be submitted to such databases. I was also very impressed by the one number you mentioned. One person performed 5,332 measurements with the pressure chamber. I think that's something we can tell our students.
I don't know that one person, I don't know that one individual measured all those. And that was in the Andes. That was in the tropics. So that involved tree climbing, I'm pretty sure. So yes, it can be done. It can be done.
But a very impressive number. So we have a first question by Keenan Gantz. He's asking, I may have missed this, but do you have a sense of how many individual plants were measured per site in the database?
Yes, I don't have an average number, but I do have that. I blanked those numbers out of the figure because there was too many numbers. But for example, in the top 10 species that are most commonly measured for chamber data, in that top 10, the number of individuals range from 65 to 406. That is not per site, though. I'd have to go back and calculate that. But these are many individuals. Yeah, and then it is many fewer individuals per site when there are instruments involved because the instruments are expensive. So there it ranged from, for the top 10 species, or top 10 number of observations, sorry, top 10 species with most observations for automated data, it ranged from 1 to 14 individuals. So
You
much
also
smaller.
mentioned sorry?
Oh,
You also mentioned the interaction across networks. Do you know how many sites are covered which belongs also to the subfluxnet sites that are still ongoing, or the fluxnet sites where we have water potential readings, and where it then might make sense to add other traits? You also know the limitation of the excited functional trait database, that we, for a given site where we really perform the measurements, maybe have access to further traits and data sets.
so yeah, we didn't. Okay, so all these networks are all a little bit different, which is one of those things. But so SAPFluxNet, to my knowledge, isn't like an active continual set of measurements. So those, but we have overlap. So for the same period of time, those in the map. The highlight those bubbles that are the color, whatever, I think it was yellow, I can't remember the color, that overlap have water potential measurements and sap flux measurements for the same time period. For the eddy covariance of the same thing, there we have a table that has the code, so you can go match it up yourself. Eddy covariance is ongoing, so I cannot say whether the water potential measurements have been ongoing, but there is at least overlap between the water potential and the flux measurements. XFT is even more different. XFT is like a really simple, okay, it's not simple. The data certainly are not simple to get, but this database structure is a table. And so it's just one big table. It's not different. It's not a relational database. It doesn't, to my knowledge, include as much site information. It's more species information. Like here's a species, submit, this is the citation for the species. Here's the average for this kind of oak or that kind of pine or something. So you can, yeah. And that's how we made the figure, comparing the minimum water potential from the two databases, just by species. It was not by site. So it is a little bit custom depending on what you're trying to overlap. I do think it is useful for thinking about, oh, this, if you have a site that has a lot of these things but doesn't have water potential yet, or maybe someone does have water potential but they haven't submitted to the database yet, that'd be nice to know about, if they're willing to share.
Tommy, before I read out your question, you want to go ahead yourself?
Right. Yeah. So thank you for this impressive talk. I think very interesting for us in the International Tree Mortality Network. Are you planning to test links between leaf water potential measurements and mortality risk in trees?
I'll be happy to field that one. I think it's a great idea, and perhaps if I find the time, but if somebody else thinks it's a great idea and wants to go for it, you totally should.
You should, basically.
Yes, you should.
Yeah.
Our funding from NSF is not, it's funny, they call it a research coordination network, but it's really the coordination part that we are supported to do. So we don't get to support students or postdocs to actually do the synthesis. And I really want to emphasize that there is no queue for who's going to write this study or who's going to write that study or anything like that. And even if multiple perspectives were trained on the same question, I think that, multiple answers, could be interesting to see. So again, if anybody is inspired, please do it. I do really like the idea of applying that extreme value theory to understand better minimum water potential, and also my own personal interests are centered often on PPD, so maybe working in that angle, but not anytime soon. That's the answer to the question. So others should feel free to dive right in.
Thank you. Gina, do you want to ask your question or?
Yes, I mean, it was a great talk, and it's really humbling to see how much work and passion you put into the PSI network. I mean, it's incredible, really. So compliments. And I'm really looking forward for actually digging into the database once it's going to be available and maybe trying something out more specifically about the dynamic vegetation modeling and drought-induced tree mortality. So my question goes towards that direction. So how could the PSI network type water potential time series best be used to parameterize drought mortality in dynamic vegetation models? What do you think? I mean, what could we try out? Do you expect for instance, species-specific hydraulic thresholds we could be using, or trait-based vulnerability curves, or emergent metrics, I don't know, like seasonal minimum water potential, critical water potential.
Yeah. I mean, I think all of those, there's a lot of ways you could approach it. When I think about the modeling aspects, on the one hand, there has been an awful lot of theoretical development to incorporate print hydraulic schemes into models, but they are all a bit different. And historically, we have not had the right data to test if this formulation performs better than that one. So that is an obvious sort of objective-oriented study that could be enabled by, the availability of more water potential data. Personally, I think that there are open questions about how well the models capture VPD-driven fluctuations in water potential and thus mortality risk, which the automated data are particularly well suited for addressing. And again, I'm not a modeler, so this is my very naive understanding, but when you think about how the traits, the traits are specified in these models, sometimes some coordination is assumed. For example, you might assume a species that is less stringent with its water regulation, more anisohydric, however you want to describe it, may also, necessarily have xylem that are more resistant to embolum, because why would they adopt such a risky strategy if their xylem are very sensitive to failure? I think that there's information in PSInet that could also allow us to test some of those assumptions that might guide the parameterization of the models.
Yeah, I'm also not a modeler, full disclaimer there, but I think especially the continuous time series, I think are more in line with the time steps of these kinds of models. And maybe this is pie in the sky because there's so many other factors that affect water potential. But one thing is like kind of thinking about the emergent properties, like some of these traits that are more static, like are there that we think of as more static, or we grab from databases that are treated as static, is there kind of temporal evolution in those in response? So not just that the water potential responds to VPD, but the water potential regulation and those parameters also respond in some way, maybe to VPD or soil moisture or drought. So.
Yes, thank you. I might have missed it, but are you planning, or do you provide common protocols? As if just taking undisturbed soil samples sounds easy, but there are pitfalls there. And then measuring water potential sounds easy, that there are pitfalls. And I think it would also be great for the community if we just have a platform where we have Prometheus Wiki, of course, but now we have moved beyond. You list all these automated measurements, and there are so many pitfalls just how to install them correctly. Do you have already, or are you planning?
That's a great point. So what we have is, we didn't really touch pressure chamber measurements because Cecilia Rodrigo Dominguez and her co-authors did such a great job with that. Anna Maria's paper that just came out does present like best practices for automated measurements, but it does stop short of a protocol. So there is definitely an opportunity there. The Riash paper that is under review right now will, in its final form, have the protocol as like SI, but not standalone. So that's a good opportunity for us, Jess. Maybe a new tab on the website, collect some protocols. Right now the thinking has been they'd be attached to the papers, but I could certainly see a case for them to be standalone. And also with all these early career training workshops, PhysVest is coming up, also Flux course is coming up, and that's more ecovariance, but publishing protocols would be a great service. You're right.
And also community feedback, how the devices are working. So I know that people are now scared to use fluoropulse because many mentioned they might work for a given time, but I've also heard if you know how to handle them, they can produce great data for quite a long time.
I think that's where it's not. I think a protocol would be fantastic. I would love a protocol on how to use some of these things myself. But there's just so much kind of species specific. At least I will speak for the continuous water potential sensors that are inserted or appended to living tissues. There's just so much variation of which species you pick, how negative they go, how their wounding responses to being cut or cored into. That I could also see the challenges of writing it. Just because it worked, and this happened with the cyclones, it worked for me beautifully. I mean, it was hard, but it worked beautifully on the species I happened to have outside my front door, and other people could not, like Kim, for example, could not get the psychrometers to work on eastern trees without a lot of wet kind of infiltrating into the sensor area. So yeah, I think we're just still in the nascent phases of some of these things. I think as there's some encouragement, maybe some of this paper with best practice, more people are willing to try to have make some guided decisions on what to try. I think we could get there. But I think as the early first step, it is not, especially since we're doing outside on living tissue, this is not something that we harvest back to the lab that's like really standardized and like controlled conditions. There's just a bit more variability there. Yeah, but I could see how research like that would be really welcome for people
I
starting.
realize, yeah, we're out of time, but we could mention that we do have a Slack channel devoted to continuous water potential measurement, which is open to everybody. And I don't know if you can figure out how to access it from the PSInet website, but hopefully, or if not, you can shoot us an email. But that has been, it's activity ebbs and flows, but it's a real thing, and you can post questions.
You can ask for sure.
Perfect. Good. I think we've passed an hour. So once more, thanks very much. Very impressive. And I'm sure that those that didn't manage today will take the advantage of watching it later on YouTube. So our next speaker in June will be Paolo Bernardino. And then stay tuned. Thank you very much.
Thank you.